Good
Good
evening.
morning.
Good evening, sir.
Good evening, sir.
Audible?
Good evening, sir.
Good evening. My voice is audible.
Yes, sir.
Yes, sir.
Okay. We'll start our class within 2 minutes.
Detta kan till
Okay, we'll start our class. I'm sharing my screen. Screen is visible.
Yes, sir.
Okay. Today we'll discuss about our last unit, that is disorganization and communication. My voice is audible.
Yes, sir.
Okay. First, what is tissue organization? Tissues are organized a group of cells with similar function, and their structure and communication are crucial for overall body function. The four main tissues are epithelial tissue, connective tissue, muscle tissue, and nervous tissue, which play a vital role in communication and coordination. From cell will divide to form a group of uh, to form a group of cells, and from that group of cells the tissue will be produced and tissue will give rise to a organ. If you go to the different class of tissue, there is four types of major tissue in our body. One is epithelial tissue. In epithelial tissue, it forms lining and covering of organs and body surfaces. All the covering and lining of the animal cell is uh, made up of epithelial tissue. Like if you see the picture, this is the epithelial cell lining of intestine. These cells are called epithelial cells. And if you go to the second one, that is connective tissue. Connective means which connect with different cells. If you go to the connective tissue, it provides support. First important point, provides support, connects different tissues and include tissues like major connective tissue is cartilage, bone and blood. Blood is a good connective tissue in our body because it connects through our body from head to tail. And if you go to the third one, that is muscle tissue, which is responsible for movement, including voluntary skeletal muscle and involuntary smooth and cardiac muscles contractions. Because this muscle tissue helps in movement or always contraction and expansion manner like involuntary in nature. If you go to the last one, that is nervous tissue, it transmits. It, before we also discuss about this nervous tissue and also neurotransmitter. Here, nervous tissue transmits different electrical signals. ଆମେ ପଢିଥିଲି we have discussed about two types of signal. That is one is chemical signals and another one is electrical signal. In this nervous tissue, it produces electrical signals and forming the communication network of the nervous system and includes neuron and different glial cell in our brain. If you go to the tissue organization and communication, how they are communicate with each other. Here in this picture, you see different tissues are there. This is epithelial tissue, and these are called connective tissue. And these circular fibers, longitudinal fibers, these are smooth muscle. These are muscle tissue, and these are called epithelial cell sheet. These are each are communicated with each other. If you go to the organization, first one is cellular packing. The major example is cellular packing. The way cells are packed together influences communication, growth, and structural support of tissues. The all the cells or tissue, they they are well communicated with each other during the growth period or during the any signaling molecules or any support required. If you go to the second one, organization is stem cell communication, because of if some tissues are damaged, here suppose here some tissues are damaged, this tissue will be repaired by this stem cell. This stem cell communication also important. This tissue architecture and the position of the stem cell within a tissue influence how they communicate with their environment, impacting tissue homeostasis and repair. Homeostasis is very important in tissue because if some part of the tissue is damaged, this stem cell will communicate at that part and it will heal that part. And this is the major communication and organization. And third one is nervous tissue communication. Previously also we discussed about this nervous tissue communication, and in nervous tissue it facilitates rapid internal communication through electrical impulses and chemical signals, enabling coordination and control of different body functions. Then the last communication is intercellular communication. This intercellular communication within a tissue communicate with each other through various mechanisms like epithelial cell also communicate with connective tissue. This connective tissue also communicate with this smooth muscle or muscle tissues, and this muscle tissue also all the tissue also communicated with also nervous tissue. These are called intercellular communications with various mechanisms and including chemical signal, physical contact and coordinate with their function. Because without any communication, all the all the different tissue like epithelial, connective and muscle nervous cannot work. They will work together. And this is very important for our tissue organization and communication. After the tissue organization communication, Then next one is extracellular matrix. Then what is extracellular matrix? Extracellular matrix is a complex network of proteins, glycoproteins, proteoglycans, and other
Sir,
biomolecules.
one question, doubt,
Yeah.
sir.
Yes.
Sir, nervous tissue, what is the importance and what is the role of nervous tissue in our body? Nervous tissue
Nervous
importance and
tissue.
its a
Okay.
role.
No. Nervous tissue play a major role in faster signaling process because all the faster signals like our blinking of eye or any uh, physical contact or any stimuli, external stimuli or environmental stimuli, all are fast gathered by our nervous tissue and it will gives a fastest signal. And according to them, our cell will, according to them and our cell will give a response. So nervous tissue is the major tissue in our body.
This nervous tissue present in our body in every part, any particular part it is present.
No, no, no. Nerve cell is there, always there. Any of our cells have been connected with a nervous tissue because it will connect to the brain and it gives our a signal to response. So nervous tissue is very important. Okay,
Thank you.
my voice is audible.
Yes, sir.
Yes, sir.
Yes, sir.
Yes, sir.
Okay,
Yes, sir.
okay. The next one is extracellular matrix. Then what is extracellular matrix? Extracellular matrix is a complex network of proteins which comprised of glycoproteins, proteoglycans, and other biomolecules that provide structural and biochemical support to the surrounding cell. The major function is the extracellular matrix is support of our cell from extracellular environment. It is present in all tissue and plays a crucial role in maintaining tissue integrity, cell signaling, and homeostasis. Because these extracellular matrix protect our cell-to-cell cohesion, cell-to-cell support, cell-to-cell signaling. We also read about how this extracellular matrix will give a different signaling process and maintaining the tissue integrity also how they are binding with each other, each cell how binding with each other, and these are important things of this extracellular matrix and how they signal with each other cells to give rise any integrity of different signaling molecules and which also maintain the homeostasis balance. Then if we go to the composition of this extracellular matrix, and this extracellular matrix as we discussed contain different fibrous proteins, these proteoglycans and some glycoproteins. In this extracellular matrix also some signaling molecules also there. And if you go to the fibrous protein, this is the structural component. This is the major protein that is the structural components of our extracellular matrix. And it provides strength and elasticity to the ECM because elasticity is the major term here used because if two cells are collide with each other, then the cell will be damaged. So the strength and elasticity is required in the extracellular matrix during the tissue formation or any other cell to cell signaling. The fast fibrous protein is collagen, and this is the most abundant ECM, extracellular matrix protein, and this also provide a tensile strength because different types of collagen molecules are present, like for an example, collagen 1, collagen 2, and collagen 3. These are the major abundant protein. Maximum protein of the extracellular matrix is collagen protein. If you go to the elastin, it gives a tissue flexibility and elasticity in lungs, arteries, and skins. Because you pull the skin will be not affected. It will be flexibility. There is a flexibility in your skin, and the elasticity also maintained in also skin. Then if you go to the another protein, that is fibronectin, this is a glycoprotein. What is the role of glycoprotein? The maximum glycoprotein are helping in our signaling processes, and that uh, and also that helps in cell adhesion, like cell to cell adhesion, how they cell with adhere with each other, and also migration, during the migration process, how the cell will be migrated. And last one is laminin, because we also discussed about laminin during our nuclear membrane protein, and this is a major component of basal lamina and crucial for the cell differentiation and adhesion. This also, this hydronectin and laminin, these are major protein required for cell adhesion, and collagen and elastin provide the collagen provide tensile strength, and elastin provide the flexibility of our this extracellular matrix. Then if you go to the second one, that is proteoglycan, they these molecules trap water and provide a cushion. And in this area, the proteoglycan maximum time they trap water, and it will provide a cushion, because water body is made up of 70% water, and all the cell or cytosolic region contains water. So water molecule is required in this extracellular network. This extracellular network gives a water balance between the cytosolic and ECM. And if you go to the different proteoglycans like hyaluronic acid, this is a glycosaminoglycan that retains water and provide a lubrication. Because as we know, this is a plasma membrane is a fluid mosaic model. How this fluid is maintained in this plasma membrane, because of this glycosaminoglycan or hyaluronic acids. And next one is chondroitin sulfate and heparin sulfate. And these are most important joint function and cell signaling. These are maximum time used for this cell to cell signaling. And the last one is glycoprotein and adhesion molecule. These facilitate cell and ECM interaction. One cell, and if you go to the, for an example, this is a cell and this is the extracellular matrix. They will interact with the cell and this ECM by these two types of protein. One is integrin and another one is cadherin. This integrin is a transmembrane protein. Transmembrane protein means this is found only in the upper part of the plasma membrane, and this that link to the ECM to the cytoskeleton. This binds to the ECM and the cytoskeleton, and gathering also mediates cell to cell adhesion and tissue formation. They will bind with cell to cell, binding with different cell to cell, and to produce a tissue. And if you go to the different, like how the cell to cell attachment or cell to cell adhesion will occur, then we go for the different cell junctions part. Then if you go water cell junction, if you go to the cell junction are specialized structures that connects for an example, this is a one cell, this is another cell. They connect with the adjacent cell or link cell to the extracellular matrix, and they play a crucial role in maintaining tissue integrity. Because if they will not interact with each other, then the cell will not form a tissue, and this maintain the tissue integrity, and this junction also play a major role in communication and regulating their cellular processes. If we go to the different classification of cell junction, then cell junction can be classified into three main categories. First one is tied junction. This one is a tied junction and also called occluding junction because this tied junction having maximum number of occluding proteins, and that's why this one is called as occluding junction. And second one is anchor or adherent junctions. This is called adhere with or attach the two cell with each other. And third one is gap junction because this gap junction play a major role for communication with one cell to another, or any ions or any molecules also pass through this gap junctions. Then if you go to the first discuss about the tight junction or occluding junction. This form a seal between the adjacent cell to prevent leakage of different molecules. This will form a seal. They are made up of different occluding protein, and they form a uh, tight protein network to seal between the adjacent cell to prevent any leakage of molecules or water. And this regulate paracellular transport because movement between the cells, and maintain cell polarity by separating the apical and basolateral surface. Suppose this is the apical surface and this is the basolateral surface. This tight junction maintains this apical and this basolateral surface. And the major protein in the tight junction are occludins and uh, ZO-1, ZO-2, ZO-3, different types of zonula occludens proteins. And this is majorly found in the epithelial cell lining, intestinal cell lining, blood-brain barrier, and kidney tubules. Because this junction create like this is the intestinal lumen. Because like villi-like structure gives us a maximum absorb. So that's why this pili-like structure will be formed in intestinal wall. This is the major junction in our cell junction. If you go to the second one, that is anchoring junction, this is the adhesion. Adhesion means
Sir,
attachment
the
of
previous slide, one doubt. I have one doubt, sir.
this slide.
No, sir, the adhesion junction.
Okay,
This, yes.
okay. What is the term?
Sir, the adherence junction.
Okay.
Adherence junction.
Which
Sir,
junction?
again explain. Yes. The diagram, the second point, second labeling point, adherence junctions. Again explain, sir, this point. This word. I do not understand what is this.
Now I am discussing about the adherent junction or adhesive junctions. This is the anchoring junction. Now I will discuss about this anchoring junction or adherent junction or adhesive junction.
Sir,
Okay.
sir, what is the role of extracellular matrix in cell adhesion and signaling?
They play a major role because without extracellular matrix, there will be no formation of any tissue. And in the formation of tissue, the cell will be interact is the most important because surface tissue is maximum exposed to the blood, but internal cell are not exposed. How they will take nutrients from the cells or blood from the blood? The cell to cell interaction is the very most important. Okay, so this
Okay,
is the
sir.
major communication process.
Sir, how does extracellular matrix related to cell growth?
Okay, if a cell, one cell, then divide into two cells. If they will not interact with each other or at, they will not attach to each other, how they will form a tissue? This is a circular cell. This is a circular cell. If they will not attach with each other or they will not communicate with each other, how will they form a tissue? So attachment is required, the major requirement for cell to cell interaction to form a tissue, or cell to cell communication to form a tissue, or cell to cell signaling to form a tissue. So this is very much important because suppose one stimuli is there, external stimuli, nervous tissue sends that signal. How the cell will behave, how will think, how will produce a response. Some organ or some are gland produce some response. So this interaction should be there. Okay,
Okay, sir.
cell to cell interaction should be there. From this cell to cell interaction, they will form a tissue, and this tissue will form a gland, and this gland will secrete some hormones or any stimulants which stimulate different parts of the body. Okay, so cell to cell interaction is most important. Without extracellular matrix, the two cell will not will not properly interact with each other. Okay. Okay. The next one is anchoring junction or adhesive junction. Adhesive means the attachment with each other, and this will provide a mechanical strength and structural stability to tissue, and this connects cell to each other to a extracellular matrix. And these types of anchoring junction, for an example, one is adherent junction, and these adherent junction are made up of actin cytoskeleton, as we previously also des- described about the different cytoskeletal element like actin, microtubules, and intermediate filaments of one cell to another and stabilizing tissue. And if you go to the different adhering junction proteins, one is cadherin, one is E cadherin, N cadherin. This is a calcium dependent adhesion protein and different types of cadherin molecules. These are linked to the cadherins to actin filaments. The cytoskeleton and the cadherin molecules will support in the extra cytoskeleton will support in the extracellular matrix, and these cadherin molecules will join the support for an join the two cells. Okay, so this is the major adherent junction. If you go to the desmosome also a adherent junction, adherent or anchoring junction. This provide a strong adhesion between the cells and resisting mechanical stress, because if you hit one cell, it will uh, it will be disturbed. If these two cells are made up of different desmosomal junctions, and it will provide a mechanical stress to the two different cells by binding with different proteins like desmocolin. These proteins helps in resist or collide between the two cells, and these are majorly found in skin cell. Maximum desmosomes are in skin cell and heart muscle, that is cardiac muscle, tissues under high mechanical stress, because cardiac function is 24 hourly activated, and this is high in mechanical stress. So these protein will resist the mechanical stress, and this is called anchoring junctions.
Sir,
You can,
sir,
you
what
can
is
go.
desmosome?
This is the desmos made up of different cytoskeleton. Suppose this is one cell, this is one cell. In between this is called our extracellular matrix. In this extracellular matrix, different desmosomal protein and cytoskeletal element is there, and these one desmosome and another desmosome, they will resist with each other to a major collision between the two cells. So this desmosome is very much important in this two cell junctions. Okay.
Okay, sir.
Then another one is hemidesmosome. This anchors cell to the extracellular matrix via basement membrane and link intermediate filaments like keratin to the ECM. And in the hemidesmosome, in desmosome there is desmoglin, but in hemidesmosome, major protein is integrin, laminin, collagen. These are the major proteins. And this will be found in epithelial cells like skin and cornea and muscle cells. Then third one is gap junction. Gap junction is the major junction which communicate with for communication between the two cells, and this allow direct communication between the adjacent cell by permitting the exchange of small molecules or ions or electrical signal. If you go to this structure of this gap junction, and this will allow this is a hollow cylindrical like structure, and this will allow communication of small molecules or ion between this channel, and it will from one cell to another cell to produce any responsive signal to the another cells, and this will pass like a channel or cylindrical in structure or barrel like structures. And the major protein in the gap junction formation is connexin. This form a connection, means connection between the cells, and this is a channel-like structure. And maximum gap junction is cardiac muscle. This synchronize heart contraction, neurons. During the electrical synapse, we also see like how the electrical signal fastly transform from the presynaptic membrane to postsynaptic membrane like a channel, and this is the major example of this gap junction. And also in smooth muscle for coordinated contractions, and the major function of these cell junctions, this is a barrier function. This tight junction prevent leakage in epithelial layer like this. This will prevent the leakage of this epithelial to epithelial layer. This will give a barrier between these two cell junctions. And next one is tissue integrity. This anchoring junction provide a mechanical stability because this is the most important mechanical stabilization. And third one is signal transmission, and this gap junction allow electrical and chemical communication between the two cells, and the cell polarity maintenance, and the tight junction help establish and maintain the different cell polarity. Like this is the apical membrane, this is the basal membrane, but deciding by this tight junctions. This is the major cell junctions which required for our tissue organization and tissue communication. Till this slide, any doubt?
Excuse me, sir.
Yes.
One more doubt, sir. What will happen if the Ca2 plus ion is removed from the tight junction?
Which channel?
What will happen if the Ca2 plus is removed from the tight
CO2.
junction?
CO2.
Cu2 plus. Cu2
Ca2
plus.
plus.
Yes, sir.
Because during our second messenger concept, we also described about this Ca2 plus ion. Because this Ca2 plus ion is like the major second messenger. Okay,
Yes,
it
sir.
will give a signal to cells for amplifying the one signal to many for synthesis of different proteins. Because if
Okay,
you
sir.
remove this CO2 plus, sorry, Ca2 plus ion from this tight junctions, and this will not produce any second machine. We also discuss about this Ca2 plus ion. Okay,
Okay, sir.
find it out.
No, sir. Thank you.
So key proteins role in our cell.
Key protein role. These
So
key
key protein.
these key
How
proteins
it helps? Yes, yes.
these key proteins will form these junctions. This key protein is the major protein which produce these junction-like structures, like this, like this. This is the major channel because maximum protein, like if you go to the plasma membrane structure, and these are the this is a semi-permeable membrane. From this membrane, if protein is not there, then carrier-mediated transport will not occur because the major carrier is the protein from extracellular to intracellular or cytosolic to extracellular. The key protein is the carrier protein, which is the embedded inside the plasma membrane. Because without the key protein, will not form any junction or any signal. So key protein is the major protein which will form a channel-like structure or junction-like structure between the cells. Okay, so this is required.
Yes.
Okay, then go for the different signal transduction. How the cell will communicate with each other or any tissue with each other by this different signal transduction. If you go to the different signal transduction, first one signal molecule is required, another second one is one cell will receive that signaling molecules. If this is the cell and this is one signaling molecule, first one signal is required and this signal will bind to a cell and it will produce different transduce or amplify that signal and it will produce a response. Then what is signal reception? Now a signal molecule or a ligand. We also call this signal molecule a ligand because it will bind to a receptor and this is called ligand receptor complex. And this ligand binds to a receptor, any type of receptor. We also give some example as there, major example like GPCR, RTK, tyrosine kinase receptor, and MAP kinase receptor. These are major receptor which will respond any type of signaling molecules. After the binding of the signaling molecule to a receptor, this is called a response. This is the response of primary response. After the primary response, the signal will be transduced, means the signal will be amplified or relay to the cell because all signaling molecules relay the signal and this signal will be go to the DNA molecules and it will start the transcription process and synthesis of mRNA and that mRNA is after the synthesis of mRNA it will produce a proteins and that protein will help to to help the cell to grow or any modification cell required or any signaling molecules required or any enzyme is required. This function will be happen after the relay or transduction of this signal. First, the signal bind to the receptor, and this is called reception of the signal in the uh, uh, in the receptor, and this is called ligand receptor complex. After the binding of the ligand receptor complex, the signal transduction or relay will happen, and in this signal transduction, signal is transmitted inside the cell via this secondary messenger. This is important. Then we will discuss about this secondary, what is the secondary messenger. And these transduction will be relayed by the secondary messenger, and this secondary messenger and protein modification will occur, and then the cellular response or any response will be happen. Uh, like any hormone is required to growth of our body. If this signaling molecule is not there, then there will be no production of any hormone or any cell growth will be not happen. So this signaling molecule is very much important. Then the cellular response, the cell activates means what? Cellular response means what? This signaling or amplifying of these molecules will bind in the DNA, and in the DNA at that specific genes, and it will produce a transcription mRNA, transcript to form a mRNA, and this mRNA will produce two proteins or enzymes. Okay, this cell activates or inhibits. Also, sometimes always not activated, sometimes also inhibition, because the maximum enzyme is not required. If substrate is low, why the say after the signaling molecules the cell will produce enzymes, but substrate is low, so inhibition is also required. They will not produce so much of enzymes or lot of enzymes. So inhibition also there. After this inhibition, the signal is stopped or regulated to prevent overstimulation, which is not required, like the different polycystic ovarian syndrome. Our hormone is not required, but we eat different color food in our market or any junk foods. They will mimic the hormone, and this when hormone is not required, but hormone is synthesized. This is called overstimulation. This overstimulation causes our lifestyle factor disease like diabetic, like PCOS, like cancer, because of this stimulus. And this signal transduction is the process by which cell converts external signal. This is called external signal into a internal response, and it enables cells to respond to hormones and growth factors for growth of the cells, neurotransmitter for communication with each, and any environmental stimuli leading to change in gene expression, metabolism, and cell function. Major of our body is gene express, and this will form a transcript, and this transcript will give rise to different metabolic enzymes which the cell will be function. And if we go to the different cell signaling process, how they cell are signal, we have four types of signaling. One is autocrine signaling, which means if you go to the structure, autocrine signaling is a cell signal itself in immune cells, because sometimes immune cells gives cell itself, because some immune cells like cytotoxic T cells or any other like neutrophils or macrophages, these cells produce a signal to itself because they contain different pathogens. So kill that cell. This is called autocrine regulation. And in this autocrine regulation, it cell is secreted its signaling molecule, and this signaling molecule is bind to its receptor, and this receptor enhance to produce different hydrolytic enzymes or any other factor to digest inside the bacteria or any pathogen. This is called autocrine signaling. This is only in immune cells. If we go to the paracrine signaling, because this is signal act on a nearby cells. In the case of neurotransmitter, because we have seen by like one neuron cell is there, another neuron cell is there. In the one cell having a axon, and another cell having the dendrites. This axon and dendritic, they are communicate with each other and they form a presynaptic and postsynaptic membrane. And in this pre and postsynaptic cells, they signal with each other and they send the neurotransmitter to give rise a stimulus to produce a cell's response. Then if you go to the endocrine signaling, then this is a hormones travel through this bloodstream, and this is a to the distance target because uh, the endocrine gland in our body is in brain. That is the major gland. We also study different endocrine glands like pituitary. This pituitary secrete a releasing factor to the target organ which will produce hormones, and this hormone goes through the bloodstream and it will act. Like for an example, if a insulin, pituitary send a signal to the beta cells and beta cell will be produce insulin, and this insulin released to the bloodstream, and in bloodstream, suppose this is insulin, this is produced to the bloodstream, and from the bloodstream insulin bind to the insulin receptor of the cells and it will uptake the glucose molecules. If glucose molecule will be not uptake properly from the bloodstream and the glucose level is high, and we will call that condition is diabetic condition. Then next one is juxtacrine signaling. This is require direct cell to cell contact. This is a notch signaling in development because one cell having a receptor and another cell having a ligand, and this ligand receptor communication is required in our developmental process. And this receptor called as notch, and this is the ligand is called as sometimes delta, and we also call this delta notch signaling during our development process. This juxtacrine signaling is very much important. And then if you go to the we study about the major part, that is second messenger. This means for amplification of this signal by this second messenger. Then what is second messenger system? If you go to the cell signaling, second messenger are intracellular signaling molecules that relay the signal. As we study here, this is relay of our signal. This means signal transductions. And in this relay of this signaling, uh, from cell surface receptor to the effector proteins, different effector proteins, this initiating a cascades of events that lead to a cellular response. That means this is the signaling molecule, and this signaling molecule will bind to the receptor, and this receptor then activate one G protein, and then these G proteins will activate the second messenger. This second, what is the function of this second messenger? And this the function of the second messenger is to amplify the signal or relay the signal by and this second messenger also called as adenyl cyclase, and this will amplify the cyclic production of cyclic AMP, and this cyclic AMP will bind to the DNA molecules and it will produce a positive or negative response to a cells. If you go to the different second messenger, first you see one is cyclic AMP, a widely used second messenger involved in various cellular process including hormonal signaling. Maximum hormonal signaling is depend on this cyclic AMP second messenger. After this production of this cyclic AMP, then this cyclic AMP will bind to the DNA molecules and DNA will give rise to uh, form a protein or a peptide that is called insulin. Then, if you not amplify the insulin concentration, then in the blood, the carbohydrate or sugar will be not absorbed properly. So insulin production is important after the binding of the signaling molecules and amplification of this second messenger. Then after the cyclic AMP, another one is cyclic GMP. This is the another important cyclic nucleotide that plays a major role in smooth muscle relaxation and other processes. Sometimes cyclic GMP also like cyclic AMP, cyclic GMP also required for the second messenger. And third one is the calcium ion. This calcium ion released from the intracellular stores to the enter the cell from the extracellular environment and acting as a second messenger to regulate various cellular functions like this. This is called second messenger, and this release of this calcium ion.
Sir, one question. I have
Okay,
doubt. One question.
okay,
So, which gland produce insulin? Sometimes that gland not produce the required amount of insulin. So, any person suffering from diabetes, they take a insulin injection. I listen
okay.
about this. So, which reason this gland not produce the required amount of insulin?
Insulin is
Due to
produced.
which
Okay. Insulin is produced by the beta cell of pancreas. This will produce insulin. Suppose for an example, today is a Thursday. We ate veg meal. The required amount of insulin is produced. Then after Thursday, there is a Friday. Then we will eat sufficient amount of non-veg food. For this absorption, we stimulate maximum of the beta cells of pancreas. They'll produce maximum amount of this insulin. After like this structure, our cell undergo wear and tear. Means the cell is undergo stress because sometimes we eat a small amount, another we have take heavy meals. And like this, the insulin gland is produced like wear and tear mechanism and in stress condition. During this stress condition, the beta cell is damaged and it will not produce insulin. So for the absorption of our glucose from our blood, insulin is required because insulin bind to the insulin receptor and it will amplify the signal, and then in the cell there is a blood vesicle, that is glucose transporter vesicle is there, and this vesicle will fuse the membrane and it will absorb the glucose molecules, and these glucose molecules are internalized by the cells, and this by the vesicles, and inside the vesicle this glucose is converted to glycogen by the glycogen synthase enzymes. So insulin is required. If you will not use insulin after the destruction of this pancreatic gland or beta cells, if you not use, the glucose amount increases inside the cell, and there will be osmotic imbalance between the blood cells and the cells. If there is a osmotic imbalance will occur and the cell will be destroyed. So there is there should be maintain of this osmotic balance. So we external use the insulin. Sometimes cell produce less amount of insulin, then we use different drugs like metformin, like drug during the diabetic preliminary diabetic condition. We use different drug to absorb our absorb our glucose or stimulate to the beta cell to produce insulin.
sir,
This
what
is
precaution we take to prevent this beta cell wear and tear condition? What could we do for
you will do only
do?
by taking balanced food. Not like you will go for the one marriage ceremony, you will take heavy meal, then after that you will take small. That is a wear and tear. mechanism, and or in stress condition, they'll produce much amount of insulin. So every time you should take a balanced meal or exercise. If the
Thank you, sir.
level increases, it will produce maximum amount of glycogen. After the body require glycogen amount, then there will be no synthesis of glycogen and glucose amount will increases, and this shoot out of these glucose molecules causes our diabetic condition.
Sir, one more question.
Okay.
Sir,
So
why it is called CAMP, cyclic AMP?
this is called adenosine monophosphate. They are produced from this uh, adenosine monophosphate produced from uh, the ATP. That is adenosine triphosphate. That is removal of two phosphate molecules to produce a AMP-like structure. If this form will be not cyclized, then it will go to the like production of ATP. So this is the cyclic AMP structures, and this is crucial intercellular second messenger because a maximum time used in hormonal signaling process. This is called cyclic adenosine monophosphate. The structure is cyclized because the bond is formed between this 2 and 4 in our glucose structure, which is cyclized form because it will not produce ATP. It will in the form of AMP. Okay.
Yes, sir. Thank you.
Then then the next one is diacylglycerol. This is a lipid molecule like second messenger, which is the key enzyme in many signaling process. This is that. That's also a second messenger. This is a lipid molecule. And another one is phosphatidyl inositol 4.45 bisphosphate. This is called PIP2. It converted to PIP3, and this is also a second messenger. And this is the precursor to DAT and other second messenger which involve in various signaling pathways. And this second messenger will amplify the signal, and this is called amplification of signal and transduction of signal by this second messenger. Then we study about the second messenger. Then now we will study about the this receptor, how this receptor will behave to a signaling molecule. Then if you go to the major signaling transduction process, the maximum time the receptor is used is G protein coupled receptor. This is called GPR, and this is G protein coupled receptors are membrane protein that detect a variety of signals and trigger cellular response, and they found throughout the body. Maximum signaling is occurred through this G protein coupled receptor like blinking of eye or any visualization process or any other sunlight exposure to body to receive the photons. This G protein coupled receptor is very much important, and in this protein coupled receptor, if you see the structure of this G protein coupled receptor, this is the membrane of the plasma membrane, and this is the exterior side, and this is the cytosolic side of the plasma membrane. If you see the structure, there is a 7 transmembrane domain, and this is also called serpentine receptor because of this 7 transmembrane paths, and these all 7 transmembrane paths are hydrophobic in nature and alpha helical structure. Because if you study the in the protein, that is alpha helix and beta sheets are the secondary structure of the protein, and maximum here is the alpha helical structure, that is 7 alpha helical paths, and these 7 alpha helical all are hydrophobic in nature. Because why this is hydrophobic in nature? Because the membrane is the hydrophobic, because membrane having the lipid molecules, and all lipid molecules are hydrophobic, and they are easily attached inside the membrane due to this hydrophobic interaction with these lipid molecules. And these 7 in transmembrane pass or serpentine receptor or G protein coupled receptor having the external four domains are there: E1, E2, E3, in and E4, and cytosolic is C1, C2, C3, and C4. And this is the structure of the G protein coupled receptor. If you see the structure of the G protein coupled receptor, G protein, uh, there is a G alpha class, that is, if you see the G alpha, this is the second messenger. Here is the adenyl cyclase, and this is, so it produces cyclic AMP, and for this cyclic AMP, the response is receptor response is beta adrenergic receptor, and for glucagon, serotonin, and vasopressin production. Then, if we go to the different G protein class, and here the say, different, like we discuss about this second messenger, and this is the receptor, because this receptor is different in different conditions, and these are in sometimes this I alpha is adrenergic receptor, muscarinic acetylcholine receptor, because these are nerve cell function, and some are odorant receptor in our nose for smell determination, and some are allergenic receptor, and some are acetylcholine acetylcholine receptors in endothelial cell, and some are rhodopsin like light receptor in rod cell in our eye. These are different class of this G proteins. Then if you go to the G protein signaling, and it's so a extracellular, if the
Sir,
extracellular
what is the structure of glucose receptor? Is it similar to GPCR?
glucose receptor.
Yes, sir.
No, there is nothing for this glucose receptor. Glucose is travel through the carrier proteins uh, by the cells. There is nothing for the glucose receptor. Glucose is transferred by the carrier protein in the plasma membrane from cytosolic, sorry, extra ECM to cytosolic pad, or
So there is no receptor for glucose molecule in the cell.
glucose directly bind to a carrier protein, like as we discussed in the secondary active transport. Sodium and glucose are co-transport to the cells. There is nothing to glucose receptor. Receptor for insulin is there. Insulin bind to the insulin receptor, and it will amplify the signal to uptake the glucose. This glucose is uptake by the different blood transporter. The transporter name is GLUT. Different organ have different blood transporter like GLUT 2, 3, 4, brain having different blood transporter, kidney having different blood transporter, liver having different blood transporter. This blood
So
transporter,
insulin is received by the insulin receptor.
they will amplify in the cell, and they in the cell the amplification will cyclic AMP amplification will occur, and the cyclic AMP bind to the DNA molecule, and it will activate the glucose transporter. This glucose transporter present inside the cell like vesicle like form, and they will fuse with the plasma membrane, and in the plasma membrane they will uptake the glucose, and they secrete it inside the cytosol.
So
After,
is the insulin receptor is like GPCR receptor?
no, 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 not like GPCR receptor, not like GPCR receptor.
Okay, sir.
When we will discuss about this RTK, This is called insulin receptor. We'll discuss about that. First, we will discuss about the GPCR. First, if a extracellular hormone communicate with related to the effector proteins through the GPCR, this covalently connected lipid hold the suppose G protein beta and G protein gamma subunits to the membrane. Here, the structure hormone bind to the inactive receptor, and this inactive receptor coupled with the GTP and form a complex with G, beta, and gamma in the resting state. And when no ligand is attached to the receptor, when there is no ligand attached to the receptor, they will form a inactive state or resting state. And in this resting state, after the binding or conformation of the receptor, after the hormone binding with the receptor, this is before hormone binding, the receptor is inactivated, and after the conformation of the receptor, conformation of the receptor changes as a result of the binding of a usual hormonal ligand to the receptor. Always any signaling molecules we call them ligand, and it will bind to the GPCR receptor. And this hormonal ligand which causes GTP to be displaced from G alpha and GTP to get bound. And mm, this after the binding of the this trimeric G protein coupled, and this ligand which cause GTP to be displaced, and this GTP molecule will be displaced from this G alpha. This is the G alpha and this is GTP, and this is called uh, G G beta and gamma form, and this GTP will be displayed. If GTP will not display, then this G protein will not work properly. So this GTP and G alpha should be displaced. And this after the displacement of this GTP and G alpha, then this G protein coupled receptor and the uh, our beta and gamma subunit which activates the adenyl cyclase, or it is produce the cyclic AMP, and this cyclic AMP enhance our different signals. So this G alpha and GTP should be displaced, and this displacement of this G alpha GTP form to produce a active signal, and this active signal will produce a cyclic AMP, and then the signal continue. Then after the GPCR signaling, another signaling is receptor for tyrosine kinase. This is very important as as you say about this insulin-like receptor. The insulin-like receptor will function like receptor for tyrosine kinase. How this tyrosine kinases work? The tyrosine kinase mediate response to a large number of signals, including peptide hormone like insulin and growth factor like epidermal growth factor. Like GPCR, receptor tyrosine kinase bind a signal, then pass the message on through the series of intracellular molecules, and last of which act as a target protein to change the state of the cell. First, you must see the structure of this tyrosine kinase before the signal coming. The tyrosine kinase receptor. This is the dimeric receptor, and these are in inactivated form. After the signal coming, like insulin or different growth factor signal will come, then the dimerization of this receptor will occur, and this dimerization of the receptor activates its tyrosine kinase domain. Because if you see any enzymes that comes under kinase family, it means it will go under phosphorylation process. Kinase means phosphorylation. Then what happens when signaling molecule bind to the receptor, the tyrosine kinase? After binding of the signaling molecules to the extracellular domain, in the extracellular domain of a receptor tyrosine kinase molecule, this cause two receptor molecule to dimerize. First, they will dimerize with each other, and this brings cytoplasmic tails of the receptor close to each other, and these cytoplasmic tail are coming close to each other because they are detach the dimerization of the signaling molecule. Uh, they got a distance with each other of these two antigen, and this dimerization will help the receptor to activate its tyrosine kinase domain, and this tyrosine kinase domain having a function of will phosphoryle with each other. This domain will phosphoryle with this one, and this domain will phosphoryle with this one. Phosphorylation means activation of this tyrosine kinase, and this phosphorylation of tyrosine on the receptor tails triggers the assemble and intracellular signaling complex on the tails, and the newly phosphorylated tyrosine serve as a binding site for signaling protein that then pass the message on to yet other protein. An important protein that is subsequently activated by the signaling complex on the receptor tyrosine kinase is called RAS protein. Then this after the phosphorylation of this tyrosine kinase, this will activate the major protein. In the signaling complex, this will activate the major protein, that is a RAS protein. And this RAS protein is a monomeric guanine nucleotide binding protein that is associated with the cytosolic phase of the plasma membrane. If we see the structure, this is the this RTK will activate this RAS protein, and this RAS protein is in the form of inactive state and GTP bound form. After the GTP form, this means one phosphate is added. After the one phosphate is added, it will go to from GTP to GTP form, and this replacing GTP with GTP activate this RAS molecule, and this RAS activation activate
सर व्हाट इज रास मॉलिक्यूल क्या है?
this RAS is a protein molecule. This is a signaling cascade, and this RAS will this is a protein, and this this signaling complex activates this RAS protein. Okay, this RAS protein is in the inactive form. After one molecule of phosphate is added to the RAS protein, and it will go for GTP to GTP, and this GTP replacing GTP with its RAS GTP activated form. This is a signaling cascade because it will amplify the signal by this second messenger.
Sir, why tyrosine amino acid always help in phosphorylation?
Yeah,
Why not other amino acids?
why?
So why tyrosine amino acid help in phosphorylation?
Because
Other
tyrosine
amino acid.
hydroxyl group in their structure. So tyrosine is always phosphorylated. Along with tyrosine, some serine and threonine amino acid also phosphorylated because they also contain hydroxyl group. So this is easily phosphorylated. Okay.
So I have one doubt previous slide. What are
Uh,
the importance of this RTKs? What are the importance?
we also told that This is a peptide hormone like insulin and growth factor signaling. If growth factor or insulin is not amplified or formed, how the glucose will uptake or any growth factor like epidermal growth factor or fibroblast growth factor, how the cell will grow. This growth factor is important for growth of the cells, and the insulin molecule is really important for uptake of glucose inside the cell, for production of glycogen, for storage. This RTK signaling is required. Okay.
Okay, sir.
Then this RAS protein will next activate the MAP kinase signaling cascade, and this MAP kinase and RAS protein, the major protein, will cause the major function inside the cell, that is cell growth and proliferation of the cells and survival. Any survival is required, this RTK pathway is required. Because survival means what? If there is the scarcity of different growth factors, then this RTK is activated to produce different growth factors, because growth factor required for the growth of the cells. So this means if growth factor is there, then the cell survival chances is high. If growth factor is not there or glucose is not there, then the cell's lifespan is very low. They will degrade, easily degrade. So this RTK signaling is required importantly for this cell growth, proliferation and survival. This is the important signaling pathway. Then after the signaling process, then desensitization of receptor also required, because always signal will not on condition. This would be off. That means desensitization refers to the process by which receptor becomes less responsive to continuous or repeated stimulation by a ligand, that is a hormone, neurotransmitter or drug. This mechanism prevents overstimulation, which is not required, and maintains cellular homeostasis. When cell become in homeostasis condition, when they require, they will produce different hormones or growth factors. But you will go for the junk foods and color food. They will mimic our hormone structure. When hormone is not required, they will stimulate the production of the hormones. So when the age will be low, the female or small child getting under puberty because of this overstimulation, because of this junk foods or colored foods, or undergone PCOS, polycystic ovarian syndrome. This is the maximum cases, polycystic ovarian syndrome. Then if we go to the types of desensitization, one is homologous desensitization and another one is heterologous desensitization. This homologous desensitization occurs when prepared stimulation of a receptor leads to its specific desensitization without affecting other receptor. For an example, beta adrenergic receptor desensitization after prolonged agonist exposure. Because this beta adrenergic receptor is a G protein coupled receptor or a GPCR. If we prolong expose with this receptor, the signal is always on. So this should be desensitized without affecting other receptor. Like they will not affect other receptor like RTK or any other receptor. If you go to the heterologous desensitization, this occurs when activation of one receptor leads to desensitize of other receptor even they are not directly stimulated. Sometimes some receptor is uh, inhibited by other receptor when there is the not directly stimulate. Like sometimes we use steroid. Steroid is a steroid is a lipid molecule. This doesn't require any receptor and it will directly penetrate through the cells by hydrophobic interaction and it will go to the nucleus and bind to the different bind to the different bind to the DNA molecules and it will desensitize other receptor like we eat chicken, broiler, broiler having so much steroid molecules in the broiler because they will stimulate broiler to produce meat by eating them different steroids and that steroid molecules goes inside our body and inside our body they will synthesize or stimulated other receptor and will undergo fat. So this is the heterologous type of desensitization. And what is the mechanism? Now, receptor, because if you desensitize receptor, then you just phosphorylated that receptor by kinase enzyme like GPCR. GPCR having GPCR kinase. If GPCR kinase phosphorylate its active site, then the ligand will not fit into the receptor. Then receptor is desensitized or that if the receptor is endocytosis process, then making the unavailable, then the receptor is unavailable, then the signal will not come.
नहीं सबको ना
If
ही
you
जिपी में
inhibit or down-regulated the receptor, inhibit or any other inhibitor will bind to this receptor, then the signal molecule will not bind to this receptor, then receptor will be desensitized. Why this is required? Now this required to prevent excessive signaling and cellular damage. We require for this desensitization for not gone through excessive signaling and cellular damage. And it also modulate drop tolerance like opioid receptor,
दीदी
desensitize in chronic opioid use and also it plays a major role in hormone and neurotransmitter
Sir, what is
regulation.
the meaning of down regulation?
Please mute
What
because
is the
if
meaning of down regulation?
down regulation means suppose one protein is synthesized always in basal level like beta actin or in actin molecules because they are cytoskeletal. Yet these
चप्पल
proteins
किनी
are synthesized in our body. If you down regulate these
ଭଲ
proteins, then
ଲାଗୁଛି କେତେ ସୁନ୍ଦର
cytoskeletal
ଦେଖ
network will not form,
ଫେର ଆହୁରି
and this
ଖଟା
will
ମେଞ୍ଚେ
disturb our cells. Please
ଆହୁରି
mute.
ଖଟା
Please mute. Okay. Sometimes some pain will produce in our body. The pain receptor is activated. Then we take different neurotransmitter or any inhibitor
ରଖିଦିଅ
to relieve the pain.
ଆଉ ଟିକେ
That's
ନା
why we
ଆଜି
use different inhibitory
ଆଜି
drugs.
ଗୋଟେ ଲୋକ ରାତିରେ ଚୁରେଇ
Inhibitory
ଦଉଛି
drugs
4ଟା ରାତିରେ
ଗଙ୍ଗା
during the
ଗଙ୍ଗାଦେବୀ
ସେ
ମ୍ୟାଡାମ ପ୍ଲିଜ
ବ୍ୟାଙ୍କ
ମ୍ୟୁଟ
କରିକି
କରନ୍ତୁ।
ଗୋଟେ ଆଣିକି ଦେଲା
ଗଙ୍ଗାଦେବୀ ମ୍ୟାଡାମ ପ୍ଲିଜ ମ୍ୟୁଟ କରନ୍ତୁ। ଗଙ୍ଗାଦେବୀ ମ୍ୟାଡାମ
Sorry,
ପ୍ଲିଜ
sorry.
ମ୍ୟୁଟ କରନ୍ତୁ, କିଛି ଶୁଭା ଯାଉନି। ପ୍ଲିଜ ଆପଣଙ୍କ ମାଇକ ମ୍ୟୁଟ କରନ୍ତୁ ସାର୍।
okay.
ସାର୍ ପ୍ଲିଜ।
Okay. In suppose some patient having different pain, we have to inhibit that pain receptor to relieve the pain. We take different drugs to relieve the pain. Or what is the meaning of down regulation? Means down regulation means less excretion. Will inhibit that receptor to do
ହ୍ୟାଲୋ
less excretion. When required, they will on. When not required, they will off. Like we taking different painkillers. When required, pain will produce in our body. When required, we take painkillers to inhibit the pain. That means we down-regulated that receptor. They will not produce pain. After some time, the receptor is activated. They'll give the pain. Then you have to take in the again the painkiller to relieve the pain.
Sir,
So
painkiller
receptor is
drugs down to regulate the receptors.
what?
Painkiller drugs down regulate the receptor around the
Down-regulate the
cell
signal.
surface.
Down-regulated the signal. If you want to down-regulate the receptor, you have to inhibit that receptor or inhibit that receptor. Or sometimes what happen slowly, like in insulin cases, when insulin is not produced, sometimes the receptor is slowly down-regulated. There is no molecules of insulin. After you, so doctor prescribe you to take external insulin to bind to the insulin, and then insulin will produce uh, uh, from the pancreas or the beta cells, and this insulin will absorb the glucose molecules.
The
This is the
receptor will
mechanism.
be down regulated or signal transduction will be down regulated.
What?
In down regulation, the
Hmm.
receptor molecule will be down regulated or less produced, or signal transduction will be inhibited. What will happen?
Down regulation means what? Down up regulation and versus down regulation. If you go to down regulation, this is the reduce that amount. We reduce the number of the receptor by inhibiting them, by taking different inhibitors. Okay, we down regulated that receptor. Because sometimes pain, during the pain, the neurotransmitter is repeatedly produced or over expressly activated that receptor which produce pain. If we reduce that receptor by inhibiting that receptor or down regulating that receptor, so the neurotransmitter will not bind to that receptor. Then the signal cascade will not occur. Then the inhibition will take place and we will go for pain relief condition. Down regulation means reduction of the signal. Up regulation means increase the signal. This is the down regulation and up regulation.
The down regulation will start from the receptor, and the
Any
receptor,
anything
yes, sir.
you can down regulate it. Gene can be down regulated. Suppose one gene, suppose for an example, P53 gene. If P53 gene is down regulated, how the checkpoint will activate it? There is no chance of the checkpoint is activated. If the insulin receptor is down regulated, then what will happen? If you take insulin, there will be no binding of the insulin to the insulin receptor. Then the receptor is damaged. So, down regulation means reduce the number of receptor in the plasma membrane by inhibiting them. Okay. Suppose 10 receptor is
Okay, sir.
there, we inhibit 6, 7 receptor in the plasma membrane, only 3 receptor is activated. Means we reduce the signal. The amplification of signal is reduced. So this is down regulation. So the last one of the cell is apoptosis or programmed cell death. If you see, then apoptosis is a highly regulated process of the programmed cell death that plays a crucial role in development, immune response, and tissue homeostasis. This is the major function of this apoptotic cell death. And this eliminates damaged or infected or unnecessary cells without causing inflammation. In the apoptotic process, there is no inflammation will occur. This is called program cell death. How cell will program? How the cell will suppose one cell is damaged? How the cell will be eliminated from our body by not in the process of inflammation. In the apoptosis process, the cell without causing any inflammation. But in necrosis process, necrotic means they will form infection or inflammation and cause formation. That is necrotic. If you go for the apoptotic process, in apoptosis, cell shrinkage or chromatin condensation will occur. The membrane will break and the DNA will fragmented and formation of apoptotic body, and that apoptotic body phagocytosed by our different immune cells by without inflammation. Like macrophages will take out that apoptotic body or neutrophils, they will take out that apoptotic body. If you go to the apoptotic process, there is the two major pathway. One is intrinsic pathway and another one is extrinsic pathway. In the extrinsic pathway, the death receptor that is uh, this is activated. How this is activated? When the cell is undergo apoptosis, the phosphatidylserine serine in the cell membrane are positioned to the cytosolic side. When the phosphatidylserine serine is positioned in the extracellular matrix region, then the death domain or any death signal will activate it, and this death signaling activation activates different caspase molecules, which is the enzymes which cleaves the different cells, which cleaves the cells. And these caspases are called serine threonine proteases, and this degrades the cell proteins. And if we degrade the cell protein, and it will creates a pore inside the cells, and the water will goes inside the cell, and the cell will be dead or killed by this extrinsic process. And this extrinsic process having different death receptor, this is FAS and TNF alpha. This is called TNF alpha. This is fast ligand or TNF alpha. The maximum time TNF alpha is activated when the cell is infected with viruses, and other day signal when the cell is undergo on any stress condition or any death signal. They will activate it fast ligand activated death domain or the TNF alpha ligand activated death domain, and this death domain activates different serine threonine proteases, that is caspases, and these caspases are called protease enzyme which cleaves the all the proteins inside the cells, and it will produce small vesicle like structure, and this vesicle like structure will uh, taken up by different phagocytic cell, that is macrophages or any other neutrophils cells, immune cells. If you go to the intrinsic, that is mitochondrial signaling, if mitochondria is damaged, because if mitochondria is damaged, because we know mitochondria is the powerhouse of the cell, there is no production of ATP. Then it will secrete, when mitochondria will damage, it will secrete different protein molecules, that is BCL2 family molecules, BACs and BAC. These regulate mitochondrial permeability. This will increases mitochondrial permeability. That means the water molecule easily transport through the
The
mitochondria.
death receptor is present in mitochondria or plasma membrane.
Which one?
Death receptors.
Which one? Which one?
Death, death receptor is present in mitochondrial membrane or plasma membrane?
Death receptor present in the plasma membrane.
Yes, sir.
Because mitochondria is a double membrane system and the mitochondria produce the energy that is in the form of ATP. If mitochondria is damaged, suppose stress condition, like any proton concentration is increased inside the mitochondria, the pH will be changed and the mitochondria will be damaged. Or leakage, any leakage or damage will activate the two proteins inside the mitochondria, that is PAX and BAC. They regulate the mitochondrial condition. And these PAX and BAC will cause a mitochondrial membrane permeability, and this membrane permeability helps the water molecule goes inside and destroy that mitochondria. And after the destruction of this mitochondria, the cell is undergo apoptotic conditions. And inside the mitochondria, the cytochromes, different cytochromes are there. Cytochrome, because as you know, cytochrome also is present in the mitochondria, and this cytochrome C will activate it, the serine threonine proteases, that is caspase. This caspase enzyme then digests all the mitochondrial protein, and it will form small like vesicular form. And this vesicle will give the signal to the cell inside the cell to kill that cell, and this killing process is called intrinsic mitochondrial killing process. And this inside in a mitochondrial killing process, then the membrane blebing like this, membrane will bleb like this, and this blebing will form small like vesicle, and this vesicle then taken by the macrophages or neutrophilic cells.
Sir, what is oxidative stress?
Oxidative stress means there is imbalance between a oxidant and antioxidants. Okay, oxidants means what? Because the if hydroxyl ion concentration or antioxidant species or superoxide radicals increases inside the cells, then it will cause a imbalance between the enzyme called oxidant and antioxidant. Antioxidant, as you know, everybody taken antioxidant during the COVID period. Every you know, vitamin C is an antioxidant. Okay, you taken different vegetables for different antioxidant, because antioxidant reduces our aging process in our body. This antioxidant, if oxidant, means this reactive oxygen species or free radicals or uh, any any stress like molecules form inside the cells, and this will increases, and this will damage to our lipid, protein, DNA, as well as different biomolecules. So this imbalance between this oxidant and antioxidant causes damage to our cell, because we have different enzymes, like you know, glutathione. Everybody know about the glutathione. Glutathione is a major antioxidant which reduces the genometric compounds in our body or any other harmful signal in our body, or catalase, which cleaves hydrogen peroxide to water and oxygen inside our cell. If hydrogen peroxide increases, it will produce hydroxyl radicals, and radicals mean one lone pair of electrons are free outside the cell. It will easily attack to
So,
any
so, sir,
protein.
why it is called as oxidative? Due to oxidation, it is released radicals.
Oxidation means what? Oxidant means what? Gain. Oxidant means for what? One loss of electrons. Okay. They'll oxidant and antioxidant. Oxidant means what? That is uh, like hydroxyl radicals, superoxides. These are oxidants. They are having a lone pair of electron free outside its the same.
Sir,
So,
oxidative molecule always be oxygen molecule.
oxidative molecules means which the molecules having three lone pair of electron. Okay, any
Yes.
you can take any example like reactive oxygen species, reactive nitrogen species, or hydroxyl radicals, superoxide radicals. You can see these are called oxidative molecules or oxidants. Every day they will be easily converted to oxygen by the help of our antioxidant enzymes like glutathione, like sodium.
Sir,
is-
other molecule other than the oxygen can be a oxidative radical, oxidative stress molecule.
Other,
Other than the oxygen radicals, then hydrogen, like hydrogen, nitrogen can be radical, oxidative radicals.
yes, nitrogen species also a NO. There is also a oxygen. Okay. No, there is nothing like without oxygen. Because if you take any example like our biomolecules, every biomolecule is having oxygen molecules. In water, there is also oxygen. In NO, there is also oxygen. Then, if you take oxidative molecules, then that is oxidative molecules, that is reactive oxygen species or any reactive nitrogen species, which will produce in our.
Sir, only nitrogen can be a oxidative molecule.
No, no.
There must be a oxygen
Two
molecule combined
species,
with the other
oxygen
molecule.
as well as nitrogen. These two are oxidative molecules. From these oxidative molecules, from this oxygen, from this oxygen, the oxidative molecule will be produced that is called superoxide, hydrogen peroxide, hydroxyl radical, singlet oxygen. These are called oxidative molecules. Sorry, oxygen produce oxidative molecules. If you go to the nitrogen produced oxidative molecule, that is nitric oxide or peroxynitrate. These two are called nitrogen related oxidative molecules. Okay.
Okay, sir.
Then if you go to the control and control group, apoptosis is called programmed cell death. That is a control group. Sorry, control cell death. And if you go to the necrosis, this is called uncontrolled. Why uncontrolled? Now necrosis is form of uncontrolled and accidental cell death caused by extreme conditions such as infection, toxins, trauma, ischemia, or inflammation. Because if you are having a wound and that wound is affected by different bacteria or pathogen, it causes inflammation. After some days, this inflammation causes pus in that wound, and this pus will
Sir,
cause.
inflammation is a process or it's a symptoms?
Inflammation is a process. Inflammation means what? Inflammation means in the infection area there is a immune reaction in that area between the pathogens and in our immune cell, and this cause inflammation, and this inflammation causes a pain to our that area. Okay.
The
Unlike
inflammation will lead to symptoms, different
huh,
types of symptoms.
infection
Okay,
always
sir.
lead to inflammation or any large injury like breakdown of your leg or breakdown of your bone. This also causes inflammation because in the breakdown area the swelling occurs. At that swelling part, the immune cell will react. and in the immune cell will react to their cell swelling area, and this also causes a pain and inflammation. But if you in the inflammation part, if infection is there, that means this will cause a pus in that area, and this is the death is necrotic death. Means ତାକୁ ଆମେ କଣ କହୁଛେ ସେ ଜାଗାରେ ପୂଜ ହେଇଯିବ କି ସଢିଯିବ ସେ ଏରିଆ, that is necrosis. In the necrosis process, the cell swell, that particular area will swell. In the time of pause, the cell that portion is swell, and after that the membrane will rupture and inflammation upon resulting in tissue damage. Sometimes that portion will be also damaged, and this is called unregulated or accidental. It will not always um, regulated condition. This is unregulated condition, and this is also caused by external factors like toxins. If a toxin is there, sometimes biting of bee, this releases toxin, and that particular area is inflamed, and this is called a inflammation like conditions or infections, or a particular area, if particular area is infected, or trauma means injury, or ischemia, or any injury conditions like that. The cell swell occurs, membrane rupture, and organelle also damage that particular part, and leakage of intracellular contents triggering inflammation. Sometimes leakage also occurs because the if the inflammation will rise, and it will increase the size of the inflammatory part, and it will after that it will rupture, and it will also infect the side cells, and this called and this is always pathological condition and leading to tissue damage. If you go to the causes of necrosis, physical injury, burns, radiation, mechanical trauma. If you go to the chemical toxicity, some taken poisons, drugs, or any other heavy metal exposures. If you go to the infections, bacterial, viruses, fungi, and different cytotoxic effect, then ischemia means one part is lack of supply of blood leading to oxygen deprivation or hypoxia and damage to the particular part. If we go to the immune response or excessive inflammation causing cell damage, also excessive inflammation also causing cell damage, as you know during the COVID period. During the in the COVID, when the virus load is increases, there is a inflammation by cytokine storm between the virus and the cytokine of our immune cells, and it will cause a heavy inflammation, and this inflammation damage to the lung cell, and the lung oxygen capacity is lower, and this will damage to our cell or tissue or our lung organs. If you go to the difference between the apoptotic and necrotic death, if you see the apoptotic structure, the membrane will bleed, and after the membrane will blood, it will form small like vesicular form, and this vesicular form will be absorbed by our immune cells. But in necrotic, after the bleeding, the cell is uh, cell membrane is destroyed, and the release of different internal organelle parts or different enzymes, and it also affects the neighboring cells and or neighboring tissue. This is very dangerous conditions. Or if the inflammation is or the necrosis is very small, then it's okay. If it is very large, then it will be very serious or fatal, because infection will occur rapidly. Then if we go to the our last part, that is cell survival or mechanism, in the cell survival and mechanism, in the cell survival and mechanism, because cell survival is very important, because nature always select to survive the each cells. The survival is a fundamental biological process that ensures the maintenance and function of living organisms, and it also involves a balance between pro-survival and pro-death signal. If repaired occur properly, like we study about in G1 phase, G1 to S phase, always P53 is activated, and this P53 protein is in between the pro-survival and pro-death signal. If the DNA is properly checked out the mutation or error, then it will go for the repair process. If repair will not properly occur, then it will go for the day signal. If repair will occur properly, then it will go for the survival mechanism. That means that allowing cell to adapt to stress or repair damage process and avoid premature death. This survival is controlled by multiple mechanisms of signaling or environmental factor or different metabolic processes. If you go to the mechanism of cell survival, for cell survival, growth factor is there, very very important, because growth factor, without growth factor there will be no chances of survival, and this activate pro survival pathways like this pi3k or akt. This means the rtk signaling pathway, or mapk, this is also rtk signaling pathway. This promotes growth and inhibition of apoptosis, and this drives cell proliferation and survival. Proliferation means increase of cells. If we go to different survival proteins, then we go for BCL2 family. This is a pro survival. It will inhibit this, uh, inhibits or halt in between the pro survival and pro death signal. And if you go to the, as we discuss about this DNA damage response and repair, P53 is the major protein. And if you go for the P53, go for the different DNA repair mechanism. One is non-homologous rejoining, homologous repairing, and base excision repair. Because any base is excised, then base in the base excision repair or homologous recombination or non-homologous rejoining. These are three repair mechanism process to maintain the genome integrity and the genome is properly repaired. As we discussed about previously, the oxidative stress regulation, these cells counter attack with oxidative damage using antioxidant defense systems, such as if the oxidant, that is oxygen derivatives or nitrogen derivatives, radicals or different oxidative molecules will be produced. This will be neutralized by a defense mechanism, that is antioxidant defense mechanism. One is superoxide dismutase, means this enzyme having a power of oxidation and reduction mechanism. This is called dismutase, means oxidation and reduction. This will normalize the oxidative molecules and catalyze. The major function is it will convert the hydrogen peroxide into water and oxygen. And glutathione peroxidase, this is the major enzyme which clears the different xenobiotic exposures. And this oxidative stress pathway is always activated by NRF2 pathway, and this is also a RTK signaling pathway. And if we go to the autophagic process, as we previously
Sir,
described,
glutathione peroxide is present inside the cell or it is provided from outside? Outside.
no, inside the cells, glutathione present inside the cells.
Every cell have it.
Every cells have amount of certain amount of glutathione. Glutathione is a good buffering system, and glutathione also reduce the toxic detoxification of our body. If we go to the autophagic process, in autophagic process, the cells remove different damaged cell organelles and proteins via different autophagic
Sir,
mechanisms.
can we take it from outside, glutathione?
Yes. Glutathione also we take it from outside. This is very costly enzymatic process. As we know, glutathione therapy, people are taking glutathione therapy for fairness. If you take the example of Kajol, the heroine, from the she's black in color, but she is taking different glutathione injection. This glutathione inhibit the formation of melanin pigment in our cell because it inhibits the formation of pigment. This is a
Then is
good.
there any side effect
This is
in
no, there
taking
is no side effect.
glutathione?
Why there is no side effect? Because glutathione is a good buffer system. Buffer system means it will neutralize like in acidic condition or in an alkaline condition. But glutathione is a very costly enzyme. It will starts from 3 to 4 lakhs, and it will you will take in the interval, meaning 6 months or 3 month intervals.
Then how does the cell receive the molecule glutathione molecule?
Glutathione is glutathione. If you go to the glutathione structure, is a three amino acid structures. The glutathione having three amino acids. So these three amino acids are having buffer capacity. So there will be no problem taking glutathione.
Sir, it is directly received by the cell, or it goes to the bloodstream,
Ah,
then it goes to the.
and observed by your cells. Okay, then if you go for the also cell survival signal, as yesterday we also described about the different cell cycle checkpoints, that is cycling. Different cycling is there because this cycling checks the cell growth and cell division during the cell cycle process, because every G1 to S, S to G2, G2 to N, every cyclin and CDK is there, and this cyclin and CDK check all the stages of our cell proliferation or repair mechanism. These are the major cell survival mechanism. Okay, any doubt?
Can we get this slide or PDF?
Yes, yes, I will share the video. Doubt करो ठीक है.
Sir,
Hmm. तो क्या रोची को voice is not audible.
गुड इवनिंग सर.
हाँ.
सर से जो टाइट जंक्शन ର ଆଉଥରେ ଟିକେ ଦେଖେଇଦେବେ ସେ ଡିସପ୍ଲେ ଟା ଆଉଥରେ ଟିକେ ଦେଖେଇ ଦିଅନ୍ତୁ ସେ ସ୍କ୍ରିନ୍ ଟା.
को कौन डाउट ଅଛି? कौन डाउट ଅଛି को?
ନାଇଁ ସାର୍ ମୋର ଡାଉଟ୍ ନଥିଲା ଏଇ ସ୍ଲାଇଡ୍ ମାନେ ଏଥିରେ ସବୁ ପାରାଗ୍ରାଫ ୱାଇଜ ଦେଇଛି ତ ଆପଣ ଯୋଉ ସେଇ ସ୍ଲାଇଡ୍ ଦେଖେଇଥିଲେ ସେଥିରେ ଗୋଟେ ପଏଣ୍ଟ ୱାଇଜ ଲେଖା ହେଇଥିଲା ସେଥିପାଇଁ ମୁଁ ନୋଟ୍ ଡାଉନ୍ କରିଥିଲି ବୋଲି ଆପଣ ପୁଣି ଦେଖିବା ପାଇଁ କହିଲି.
अच्छा अच्छा ठीक है. ଆଉ କାହାର ଡାଉଟ୍ ଅଛି को?
ସାର୍ ଆର ନେକ୍ସଟ ସ୍ଲାଇଡ୍ ଟା ଆଙ୍କରିଂ ଜଂସନ ନା?
which one?
ଆ ପର ସ୍ଲାଇଡ୍ ଟା ଆଙ୍କରିଂ ଜଂସନ ଅଛି ନା?
Yes, anchoring and gap
Gap
junction.
junction.
So please say something about KD value of the receptor.
Which one?
KD value of the receptor.
KD value of receptor means how the receptor bind to a ligand, because receptor ligand binding is very important. Uh, if the concentration of ligand is increases, what will be the KD value? If the ligand concentration is increases but receptor is lower, what is the KD value?
Sir,
Suppose a receptor and a ligand is there. Okay, a receptor is a hydrophobic molecule. Okay, the ligand should be
Hydrophobic.
ligand कौन होगा दरकार? ligand जदि hydrophobic है receptor कौन होगा दरकार? जदि ligand receptor ର interaction न हो ताले को टाइप रे interaction न हो? Hydrophobic means जानिछो तो? What is hydrophobic? जानिछो ना जानिनो?
Hydrophilic, hydrophobic.
हाँ hydrophobic means what?
Hydrophilic means love for water. Hydrophobic means fear of water.
Very good. Hydrophobic means devoid of water molecules. Means
Sir.
lipid is a hydrophobic molecule.
Sir.
Okay. जदि गोटे ligand hydrophobic हेइतिबो ताहाले तमर रिसेप्टर ता जदि हाइड्रोफोबिक हेइतिबो ताले बाइंडिंग ता ताकर हाइड्रोफोबिक इंटरेक्शन होबो. ओके.
Sir.
जदि हाइड्रोफोबिक इंटरेक्शन होबो ताले they will bind tightly and the KD will increases. Okay.
Sir.
Sometimes some protein have small number of hydrophobic molecules. Suppose one protein is having 4 hydrophobic molecules, but some protein having 8 hydrophobic molecules. When it will bind to the receptor, then the KT will be what? Kahar KT increase kari jibo? A protein having 6 number of hydrophobic residue, B protein having 8 number of hydrophobic residue. তাহলে কাহার binding affinity high হবে receptor সহিত, কারণ receptor is hydrophobic, কাহার binding affinity ଅଧିকা হবে?
Sir, the protein with 8 residues.
हाँ, যার 8, কারণ তা পাখে hydrophobic moiety ଅଛି ଅଧିକା ଅଛି, তাহলে ইয়ে কোন না বেশি tightly binding to the receptor. And if the receptor will be tightly binding to the receptor, the KD will increase. KD means what? Dissociation. Or tightly bind means केमिति bind करूचंती गोटे ligand और गोटे receptor complex. ता ऊपर डिपेंड करू. तो तमर ता ऊपर डिपेंड करिवो ना तमर जो ligand molecules ता अछि से जदि ता receptor सहित bind करूची. Suppose यदि देखिबा इठी ओके एटा गोटे हला मोर ligand टा एटा हला receptor. ए receptor टा हला hydrophobic in nature. ए जो ligand टा अछि एई जो लाइगैंड रे जदि हमर हाइड्रोफोबिकनेस बेसी रहिबनी ताहले आर केडी विल डिक्रीज जदि आर देयर हाइड्रोफोबिकनेस अधिका रहिबो then the KD will be increased. Means
increase का
it show that there is a strong association between this ligand and the receptor complex.
receptor
Okay,
sir
this is the way
Okay, sir.
measuring the KD value. This measures the affinity of ligand to its receptor. Okay, this is called KD.
sir
How got it out with you?
sir
So,
Uh,
so can we get the recorded videos of this class?
Please contact to your coordinator, sir, or local of your coordinator for this video.
Okay, sir. Sir, and we haven't received any PPTs of this class. The last three classes also we haven't received. So, how can we get it?
What?
So, PPT.
PPT. I
Yes.
also sent all the PPT
So, we
to
haven't received
friends.
anything.
If you did not get any PPT, please
No.
message me and I will share all the PPT. Okay.
Okay, sir.
सर सर जदि आउ किछि डाउट हला मैं तो भलो देखिनु देखि सल्ला परे किछि डाउट हला आपन को टाइम रे हम पचारले
हाँ
क्लैरिफिकेशन
हाँ
पाई परिबे
पचार ମୁଁ କହିଦେବି କିଛି ଅସୁବିଧା ନାହିଁ।
नहीं आपण सब समय तो व्यस्त थिबे को समय टिके फ्री थिबे जोडै पचारि परिबा हम
ଆପଣ 8ଟା ପରେ ମୋତେ ପଚାରି ପାରିବେ।
राति 8टा पर सर
ହଁ ହଁ 8ଟା ପରେ ମୋତେ ପଚାରି ପାରିବେ।
सर
ଆଉ ଯେତେବେଳେ ବି ପଢିବେ ଆପଣଙ୍କର ଯୋଉ
सर
ଦିଆ ହେଇଛି ବୁକ୍ ଦିଆ ହେଇଛି ସେତେବେଳେ
सर
ଏଇ ମୋ ସ୍ଲାଇଡ୍ ଗୁଡାକ ରଖିବେ ସେଇ ଟାଇମରେ ସାଇଡ୍
सर सर
ହେଲେ
सर
ଆପଣଙ୍କୁ ବେସିକ୍ ଜାଣିଲେ ଆପଣ ସେଇଟା ବୁଝିପାରିବେ ନହେଲେ କିଛି ବୁଝିପାରିବେନି।
ନାଇଁ ସାର୍ ବର୍ତ୍ତମାନ ସୁଦ୍ଧା ତ
ଇଏ ବି ଯଦି ଆପଣ ଜବରେ ଥିବେ ବି କୋଉଥିବେ ଯଦି ଆପଣ ବ୍ୟସ୍ତ ଥିବେ ବି ତ ସେଇ
ସାର୍
ଅନୁସାରେ ଆପଣଙ୍କୁ
एक्चुअली ଆମେ सर्विस सबुडा सर्विस होल्डर साର୍ तेणु सबुडा तो टर्न बुझी होनी आको डीपली देखले ही हमें बुझी परिबो आको इंटरप्रेट करिवो आपनका स्लाइड सहित बही
ହଁ
को
ହଁ
तेबे
ମୁଁ
जाई
ସବୁ
50%
କଭର
बुझीबु
କରିଛି
सर
କିଛି ଆପଣଙ୍କୁ
सर
ଅସୁବିଧା ହବନି ଆପଣ ଇଜିଲି ଦେଖିଲେ
सर
ବୁଝିପାରିବେ।
हाँ सर तो ସେଇଥିପାଇଁ ସବୁ ତ ସାର୍ ବୁଝିବନି ଯେହେତୁ ଟେକ୍ନିକାଲ ୱାର୍ଡ ତେଣୁ ଆପଣଙ୍କୁ ପଚାରିକି ଟିକେ
No,
ଡିଷ୍ଟର୍ବ କରିବୁ ସାର୍
then you can take any
ସାର୍ ସାର୍
WhatsApp message or any
ସାର୍
call me after for your any doubt.
ସାର୍ ओके सर थैंक यू
ଆଉ କାହାର କଣ ଅଛି doubt?
ହ୍ୟାଲୋ ସାର୍ ଗୁଡ୍ ଇଭିନିଂ।
Good evening.
ସାର୍ ଏଇ ଯୋଉ ଆପଣଙ୍କର ସ୍ଲାଇଡ୍ ଆଉ ବୁକ୍ ଟା ଯଦି ଦିଟାକୁ ମାନେ ଇଏ କଲା ବେଳେ ଟିକେ ଅଲଗା ଅଲଗା ଲାଗୁଛି ସାର୍।
ଅଲଗା ଅଲଗା କେମିତି
ମତଲବ
ଲାଗିବ ସେମିତି ତ
ବେଶୀ
ଲେଖା ହେଇଛି।
ବୁକ୍ ରେ ବହୁତ ଏକ୍ସପ୍ଲେନ ହେଇଯାଇଛି ସେଥିରେ ବି କିଛି ୱାର୍ଡ ଟା ବୁଝି ହଉନି। ଫେର ଆପଣଙ୍କର ସ୍ଲାଇଡ୍ ରେ ବି ମତଲବ ଦିଟାକୁ କମ୍ପାରିଜନ କଲେ ମୁଁ ଟିକେ
ମୋ
ବୁଝିଲା
ସ୍ଲାଇଡରେ
ବେଳେ
ବୁଝିପାରିଲେ ନା
ପ୍ରୋବ୍ଲେମ।
ନାଇଁ ଆପଣ?
ଆଜ୍ଞା ସାର୍।
ମୋ ସ୍ଲାଇଡରେ ଯୋଉଟା ମୁଁ ଲେଖିଥିଲି ବୁଝିପାରିଲେ ନା ନାଇଁ?
ହଁ ଆପଣଙ୍କ କ୍ଲାସ ଆଟେଣ୍ଡ କଲେ ସବୁ ତ ବୁଝିଲିନି ବଟ୍ କିଛି କିଛି ବୁଝିଛି ପୁରା
ହଁ
ପ୍ରପର ସବୁ ବୁଝିନି ଆକ୍ଚୁଆଲି।
ହଁ ଠିକ ଅଛି ଯେତିକି ବୁଝିଛନ୍ତି
ହଁ
ତାକୁ ଯଦି ଆପଣ ପଢିବେ ଆପଣ
ହଁ।
ସେଇ ଯୋଉ ବୁକ୍ ରେ ଯୋଉଟା ଦେଇଛି ତାକୁ ବି ବୁଝିପାରିବେ।
ଓହୋ ବୁକ୍ ରେ ଟିକେ ଅଧିକ ମାନେ ଲେନ୍ଦି ଟିକେ ଦେଇଛି ତ ଏଇଟା ଟିକେ
ହଁ
ସର୍ଟକଟ
ସେଥିପାଇଁ
ହେଇଯାଉଛି
ତ
ତ
ମୁଁ
ସେ
ତାକୁ ଆପଣଙ୍କୁ ବେସିକ ରେ ପଢେଇଲି ନା ଲେନ୍ଦିଟାକୁ ବେସିକ ରେ ପଢେଇଲି ତ ଆପଣ ଏବେ
ହଁ ହଁ
ଯେତେବେଳେ
ହଁ।
ପଢିବେ ଆପଣ ବୁଝିପାରିବେ।
ସାର୍
ଓକେ
ଏକ୍ଜାମ ଯେତେବେଳେ ଦେଖିବୁ ଆମେ ଆପଣଙ୍କୁ ପିପିଟି ସେଣ୍ଡ କଲେ ହବ ନା?
ହଁ ଲେଖିପାରିବେ ହଁ
ଧର
ହବ
ମୁଁ
ଆପଣ ଏକ୍ସଟେଣ୍ଡ
ମୋର
କରିବେ ଟିକେ
ମୁଁ
ଏକ୍ସଟେଣ୍ଡ
କହୁ
କରିବେ।
ଯଦି ଆପଣ ଯୋଉ ବ୍ରାଞ୍ଚ ବନେଇବେ
ଟିକେ ଏକ୍ସଟେଣ୍ଡ କଲେନି କି?
ସେ ରୁ କିଛି କିଛି ଲେଖିଲେ ସେମିତି ଲେଖିଲେ ହବ କି ପୁରା ଯେମିତି ବୁକ୍ ରେ ଆମର ଅଛି ସେମିତି ଲେଖିଲେ ବେଟର ହବ କୋଉଟା ମତଲବ ଭଲ ସାର୍ ଆମ ପାଇଁ ସେଇଟା
ନାହିଁ ଆପଣଙ୍କର ଆପଣ ପଏଣ୍ଟ ଦେଇକି ଲେଖିବେ ଆଉ ମୁଁ ଯେତିକି କହିଲି ତାକୁ ପଢିକି ଯଦି ଆପଣ ଏକ୍ସଟେଣ୍ଡ କରିବେ ଟିକେ ହେଇଯିବ।
ହେଡିଙ୍ଗ ସବ ହେଡିଙ୍ଗ ଦରକାର ନା
ସବ
କି
ହେଡିଙ୍ଗ
ଏମତି
ସବ
ଥିଓରି
ହେଡିଙ୍ଗ
ଟାଇପ ଜଷ୍ଟ ଯେମିତି ନାଇଁ ନାଇଁ ସାର୍
ହଁ
ବୁକ୍ ରେ ଯେମିତି ଲେଖା ହେଇଛି ସେମିତି ଟିକେ ଲେଖିବାକୁ ପଡିବ କି ଆପଣ ଯେମିତି ଟିକେ ବ୍ରିଫଲି ଦେଲେ ଛୋଟ ଛୋଟ କରିକି ସେମିତି ଲେଖିଲେ ବେଟର ହବ ସେଇଟା ମୁଁ ପଚାରୁଥିଲି।
ହୁଁ।
ହ୍ୟାଲୋ।
ହୁଁ।
କିନ୍ତୁ ପିପିଟି
ଓକେ
ଯାକ ମୁଁ
ଆମେ
ପାଉନି
କ୍ଲାସ
ଦିଟା
କ୍ଲୋଜ
ପିପିଟି ହିଁ
କରିବା।
ମିଳିଛି ଆଉ ଦିଟା
ମତେ ମେସେଜ କରିଦିଅ ମୁଁ ପିପିଟି ସେୟାର କରିଦେବି ଆଉ କ୍ଲାସ କ୍ଲୋଜ କରିବା। ଆମେ ଯାହା ବି
ଆଚ୍ଛା
ଡାଉଟ ଥିବ ମତେ ମେସେଜ କରିବ। ଠିକ ଅଛି।
ହଁ
ଯଦି
ସାର୍
ପିପିଟି ଦେଇଥିବେ
ସାର୍
ଭଲ ହେଇଥାନ୍ତା
ସାର୍
ଟିକେ ଆମେ ନିଜେ
ନମସ୍କାର।
ଦେଖି ଦେଇଥାନ୍ତୁ ଆପଣ
ହଁ ହଁ ଠିକ ଅଛି ଯାହା ଡାଉଟ ହବ ପଚାରିବେ କିଛି ଅସୁବିଧା ନାହିଁ।
ନମସ୍କାର ସାର୍।
ହଁ ଥ୍ୟାଙ୍କ ୟୁ।
Thank you, sir.
Thank you, sir.